these juices collectively form gastric juice. Its daily secretion is 2 to 3 liters per day. The pH of gastric juice is 1.2 to 1.8. HCl forms about 0.05 to 0.3% of gastric juice. Gastric secretion is stimulated by thought of food, smell of food, chewing of food, and the contact of food with stomach wall. Third type of gland which is present in our body, that is in digestive digestion system, is intestinal glands. These are numerous microscopic glands present in the mucosa of small intestine between the villi. These are following two types. First is the crypts of Lieberkühn. These are multicellular, simple, straight tubular glands present throughout the mucosa of small intestine at the basis of villi. These secretes digestive enzymes and mucus. These have four types of cells. First is pain cell, secretes lysozyme and antimicrobial agents. Second is argentaphine cells or enteroendocrine cell, produce secretion and serotonin hormones. Serotonin inhibits secretory activity of gastric glands but stimulates salt secretion in pancreatic juice and bile, whereas serotonin stimulates peristaltic movement in intestinal wall. Third one is the goblet cells, secretes mucus. Fourth is enzyme secreting cells, produce enzymes of intestinal juice. Brunner's gland. These are compound tubular glands present only in the submucosa of duodenum. They open into crypts of Lieberkühn. They secrete alkaline, enzyme-free watery secretion. Secretion of intestinal glands is called intestinal juice or succus entericus. It is alkaline with pH 7.8 to 8.3. It contains enzyme maltase, isomaltase, sucrase, lactase, alpha dextrinase, limit dextrin, enterokinase, aminopeptidase, dipeptidase, nucleosidase, intestinal lipase, etc. About 2 to 3 liters of intestinal juice is secreted per day. The largest gland which is present in our body is called liver. It is weighed about 1.5 kg in a human body. Liver fits into the concavity of diaphragm and is attached to it by a medium vertical fold of peritoneum. The liver has two main lobes, large right lobe and small left lobe, separated by falciform ligaments. In addition, there are two very small lobes called quadrate lobes and caudate lobe. Gallbladder, it is a thin-walled pyriform sac that lies in the lower surface of right lobe. It stores bile secreted by hepatic cells of liver. Bile is carried from the gallbladder to the duodenum by bile duct. It is formed by the fusion of cystic duct from gallbladder and hepatic duct from liver. Bile duct joins pancreatic duct to form hepatopancreatic duct. This duct is dilated, forming hepatopancreatic ampulla or Vater's ampulla. Its opening to duodenum and its opening is guarded by a sphincter of Oddi. The bile duct is surrounded by a strong sphincter muscles of Boyden before it opens in the pancreatic duct. When these muscles contracts, bile juice is formed back into the gallbladder for storage. Function of liver. Liver is one of the most vital organ of the body. Its main functions are production of bile. Liver cells manufacture bile. It is a yellowish green or reddish brown watery fluid, alkaline in nature. pH 7 to 8.6 and bitter in taste. It contains bile salts and bile pigment. Sodium thiochocolate, sodium glycochocolate, and sodium bicarbonate is the bile salt. Bile pigment is the bilirubin and biliverdin. Metabolism of glucose. Glycogenesis. Excess of glucose from blood is converted into glycogen and stored in the liver cell. Glycogenolysis. Whenever needed, glucose is obtained by the hydrolysis of glycogen. Glycogenesis. Whenever needed by the body, glucose is synthesized from amino acid or fatty acid and glycerol. Synthesis of fat or lipogenesis, excess of glucose is converted into fat to be stored in adipose tissue. Beta oxidation, denaturation of fatty acid and phosphorylation of fats takes place in liver cell. Deamination of proteins. Excess of amino acid undergo deamination producing pyruvic acid and ammonia. Synthesis of urea. Ammonia produced by deamination of amino acid in hepatic cell is converted to urea, that is also called Krebs ornithine or arginine cycle. Synthesis of vitamin A from carotene and storage of vitamin A, B12, E and D. Synthesis of albumin from amino acid. Storage of inorganic substances like iron, copper and potassium. Formation of blood proteins, that is prothrombin and fibrinogen. These are necessary for blood clotting. Phagocytosis. Kupffer cells destroy dead RBCs. The bile pigments bilirubin and biliverdin are formed from the breakdown of hemoglobin. Produces heparin, an enzyme which prevents clotting of blood inside the blood vessels. Formation of RBC during fetal development. Manufacture of lymph. Liver is an important seat of lymph formation. Detoxification. Liver cells either inactivate the toxic substance like cresol, carbolic acid, etc., produced by intestinal bacteria, or it converts them to non-toxic substance. Similarly, prussic acid producing during metabolism is converted into non-toxic substance. Liver is center of heat production, osmoregulation. Liver produces angiotensinogen. It helps kidney in maintaining body fluids. The bile pigments bilirubin and biliverdin are excretory product. Obstruction of hepatic or bile duct by gallstone causes obstructive jaundice. In this disease, bile is absorbed in the blood instead of going to the duodenum, causing yellowing of eyes and skin. Second largest gland which is present in our body is called pancreas. Pancreas is diffuse lip-shaped gland. It is about 16 centimeter long and 2.5 centimeter broad. It weighs about 60 grams. It is compound gland formed of exocrine part and endocrine part. Exocrine part is formed of lobes of acini held together in connective tissue. Each acinus is formed of single layer of large granular acinous cell that manufacture pancreatic juice. About 500 to 800 milliliter of pancreatic juice is secreted every day. Pancreatic juice is alkaline, having pH 7.5 to 8.5. It contains sodium bicarbonate and enzymes such as trypsinogen, chemotrypsinogen, and procarboxypeptidase. Endocrine part, it is represented by patches of cell in the exocrine part called islets of Langerhans. These are formed of 4 types of cell. Alpha cell, these produce hormone glucagon. It induces conversion of stored glycogen into glucose in liver cell, causes release of glucose in blood and elevation of blood sugar level. Beta cell, these produce hormone insulin. Insulin promotes formation of glycogen from glucose for storage in liver and muscle cell and reduces blood glucose level. Deficiency of insulin causes diabetes. The two hormones, insulin and glucagon, are antagonistic in function. Third type of cell is called delta cell. This cell secrete somatotestin hormone which inhibits the secretion of glucagon and insulin. Somatotestin also slows down absorption of digested food by gastrointestinal tract. Pancreatic polypeptide cell or PP cell or F cell, they form about 15% of islets of Langerhans. They secrete pancreatic polypeptide hormone which inhibits release of pancreatic juice. Function of pancreas. Pancreas secretes about 1 liter of pancreatic juice which is alkaline with pH 8.7. It contains digestive enzymes and hormones. It is carried to the duodenum by pancreatic duct, also called duct of Grisang. In addition of these glands, the entire eliminatory canals has mucus glands that produce mucus. The mucus lubricates the digestive tract so that food slips through it easily. Mucus also prevents injury to the delicate mucous membrane and protects underlying cells from digestive enzymes.